Alright, everybody, welcome back to another video. This is lecture 14, the last video I'm recording for tonight because it's 10:30 and I want to go do something else, but let's get into this then, yeah? First, I actually want to make a bit of a correction to what I said before about APC and APC/C, which I think is mentioned below, if I'm not mistaken, so I won't mention it now, but that's a very important correction. I'm pretty sure I made a mistake in the last video with how I talked about APC, but I already talked about a lot of this stuff. Cancers being driven by or caused by a lot of different things. Stem cells, basal cells, cells that are actively dividing within the skin. Stuff like that. Colorectal cancer. Colorectal cancer, again, elevation of Wnt signaling leads to right cancer. So there's a bunch of stuff here. Already talked about this, but this is what I wanted to bring your attention to. This right here. I'm pretty sure I said that the APCs are the same in the last video. That is not correct. Please disregard anything I said about that. I think that was like a small comment I made, if I'm not mistaken. APC in mitosis is anaphase promoting complex, and then the APC in this is adenomatous polyposis coli, which is not the same thing, so just keep that in mind. Already talked about all of this, so I'm not gonna harp on it, but when beta-catenin is not degraded, it activates this, and then you get all sorts of production of genes that lead to division. Cool little piece of data here, study over 1,000 humans. 96% of samples had elevated Wnt signaling, so yeah. But yeah, I mean, this should make sense, right? When APC is mutated, this complex never forms, so you never degrade beta-catenin, and so you just get this consistent activation. So, yeah, this complex that degrades beta-catenin doesn't work, and so you just get a bunch of cancerous tissue forming, so. Right, so I think in this tumor heterogeneity, I'll be honest, so this set of slides, from when I went through it the first time, didn't seem like there's a lot of content that's actually worth covering, but I'll go ahead and go through each slide and point out things that I think might be important, but there's just a lot of stuff in here that I don't know how they'd exactly test you on this. Refer to Professor Kong and whatever she said in terms of if you're looking for things that what specifically is going to test you on, but this stuff's really interesting because I'm not sure how you can really test this specifically, but linear tumor evolution, we first talk with tissue that's chill, so non-malignant just means chill tissue. It's not really doing anything. We get an oncogenic hit, so a mutation hits that leads to a cell dividing more than often. So the cancer cell has a selective growth advantage, so this cell here divides more often than others. And so this cell divides more and more, and you get copies of it, basically. So you get copies of this cell. And eventually, this cell just continues to divide, and it continues to divide, and you get a bunch of cells that are genetically unstable. And so, this cell will continue to divide over and over again, gain these advantageous mutations that help it with staying cancerous and evading immune system detection. And so what basically happens is that these new clones will outcompete the old ones, right? Because the new ones have these newer mutations that are more favorable for cancer cells. And so the old cancer cells will eventually die off, but these new cancer cells will continue to thrive and reproduce. So it's just evolution, right? Evolution, this idea that the genetic composition of a what's it called, or the allele frequencies change from generation to generation, and that is exactly what is happening in this case. Branch tumor evolution is a little bit different. So with linear, it's like we get a mutation, that mutation proliferates, new mutations pop up in the new line, and those new line mutants replace the old line mutants, basically. In branch tumor evolution, we have the first mutation still. We have this additional driver mutation. And so then instead of having like just one population, we get two different populations, so or multiple different. That doesn't have to be two, but we get a propagation of multiple subclonal populations sharing common ancestors. So basically, you have many different like advantaged cancer cells that divide consistently over and over again, and you get all these different strains. Essentially, what I'm trying to get at. So. All this to say that tumor heterogeneity facilitates colorectal cancer progression. So basically, this process of mutating, replicating, and mutating again is what drives colorectal cancer progression, and ultimately is one of the things that actually stops it from being easily treatable for a lot of cancers, really. So you can see here, actually, here is a series of things that causes this. So like here we have, there's a loss of APC first, there's an activation of KRAS, then there's a loss of SMAD4 and other tumor suppressors, loss of P53 and other unknown things. So you can see here that this is like a fourfold causation of cancer here. We've got, so I mean, this already is bad enough to cause cancer, but then we add on these things over and over again, and we really are getting into the case of, this is a very complex cancer to solve. So we can see here, the question is, if it's initiated by a mutation APC, what therapy do we use to prevent it initially? And you would look for something that would prevent APC from being lost. So Wnt inhibitors is a great example of this. So like a Wnt inhibitor would maybe a competitive inhibitor for the receptor, right? Will bind and stop it from activating that pathway, perhaps. So here we have an example of this. So we have scientists actually did develop a Wnt inhibitor, and this thing actually inhibits or reduces Wnt signaling by 50%, around 50%. So it's great usage actually. So. This is a really cool drug because what it does is tankerase is the tankerase inhibitors. Actually, what they do is they stabilize axin. So axin is one of these adapter proteins that binds CK1, GSK3. So it actually stabilizes axin, allows these things to bind even when APC is muted. So it doesn't matter that APC is muted. APC is mutated. This complex will still form, and that's great because if the complex continues to form, then what that means is that we can degrade beta-catenin, and so we don't get any cancerous activity, really, which is great. This is just a way of saying that this drug worked to inhibit growth in other cancer cell lines as well, so not just in the colorectal cancer cell lines as well. The unfortunate thing about this is there are some extra issues that could come from this, and there's a thing about here with mice treated with these tankerase inhibitors were unfortunately sacrificed due to excessive weight loss, which is pretty sad, but such is the cost of research, sadly. Further analysis revealed that severe necrosis and inflammation of the small intestine. So basically, we stopped the division of these small intestinal cells, and the small intestine really does. There's a lot of division that occurs in the small intestine generally to try and keep the intestine healthy. But you get rid of that, and the small intestine is still it's a digestive organ, so there's a lot of raunchy stuff going on around there. So like a lot of acid, not acid necessarily, but a lot of things that could be considered detrimental or harmful to cells, which is why you need to constantly replace the cells over and over again. So you can probably understand why it would lead to necrosis and inflammation of the small intestine, which would lead to weight loss and eventually death. if not correctly monitored and treated correctly. A bit of a side tangent here. I usually don't go on tangents, I know. I do want to kind of point out research as just research in general, and the dangers of research and what goes on in research, because for some people it's just not the thing. Like for me, I've been lucky enough where I don't have to. For me, it goes against my personal beliefs to, kill animals, even if it is for the purpose of research. Although I obviously know that, you can't really further scientific discovery if you don't use animal models. Animal models are really important. So, basically what I'm trying to say is if you're doing research, whether it's for med school or for grad school or whatever, try and do research that you actually enjoy doing. Don't do research because some guy on Reddit said that you should do research and publish 5 papers on murdering rats because it'll make you get into med school. That's just not true. Do research you enjoy doing, and if you do research you enjoy doing, you will publish, right? You will publish, you'll produce great results, you'll produce great posters because you just like doing it. So make sure you do things you actually want to do, and don't just fall into this hole of trying to do what you think is going to make others happy, because I've had friends who have done this where they've had to drug rats, and they've had to unfortunately execute them or kill them at the end, and it's taken a huge toll on their mental health. Luckily, they're in a better place now with their new lab, but this is a thing. So take care of yourself is what I'm trying to say here. Anyways, let's forget about my little tangent there and move on to actual content. So, what is the best way of treating cancer? So, actually, early detection and removal of the polyps is the best treatment we currently have against colorectal cancer. And basically what this is just you just need to remove the cancerous, tumors, basically. So, benign... I just realized, did I mess that up here? Did I say malignant is fine up here? No, non-malignant. Okay, I was like, did I mess this up? No. Anyways, yeah. So here we have some polyps here, right? They're starting to form. So there's some level of division here, even more division, some more division, some more division. And at this point, we're starting to get into, oh gosh, this is really large. And then here we can see it's actually starting to metastasize into other areas. So at this point here, it's getting malignant, right? And so what we need to do is we need to remove it before it gets to this stage, ideally. So remove it here. If you remove it there, remove it here, remove it here, remove it here even. If you remove it, the sooner you remove it, the higher the chance of you just being happy you are. So, yeah. I think I messed this up. Yeah. This. I was on the wrong slide. But you get what I was saying. Remove it here. Try not to let it progress to this stage and beyond. Alright. I'm not going to go over this too much. Remember, RAS is the protein that stimulates, is the protein that involves in the external growth factor pathway. So this is a proto-oncogene. If you mutate it, it activates. P53 inhibits, so it's a tumor suppressor. RB, tumor suppressor. EGFR, this is the growth factor receptor. CDK is remember the thing that you guys talked about that promotes mitosis. Cyclins promotes mitosis. APC, so the anaphase promoting complex. Oh, what the heck what just happened there? That's so weird. APC, remember, this is not the, this is, oh god, this is there's a very specific slide I'm thinking of, and this is actually an inhibitor. I keep saying anaphase promoting complex. Now that I think about it, I can't even remember if that's right or not. I think it's APC. Let me double check that actually before I feel like I'm lying a lot these days. No, it is, sorry, no, it is anaphase promoting complex. Yeah. Say it's a tumor suppressor here. I can't remember exactly where this one acts, so you might have to go back and double check this for me. Someone let me know in the comments maybe where this is. I can't quite remember off the top of my head. PASH, remember, PASH inhibits smooth end um, leads to a degradation of glee, and so glee doesn't do anything, right? So pasture is a tumor suppressor. Smoothened is smoothened when active turns glee into a glee activator. APC, remember, is the one that helps with degradation, so it's a tumor suppressor. And then beta-catenin, when active, is a proto-oncogene. So there's a couple things to keep in mind. Okay, what does it take to cure cancer? Gonna be honest with you, this section is so open-ended to me that I'm not sure how useful this is, but what does it take to cure cancer? It takes a lot, right? There's not a single cure for cancer, because there's not a single cause. You have to consider a lot of underlying biology for each cancer. You have to consider specific drugs. What are their side effects? You have to design methods to target these cancer cells specifically. Therapies, like can the drug that you're making work with other drugs without killing somebody? You also have to take into account a person's genetics. Like, sometimes a drug is really bad because it takes a genetic predisposition that somebody has and makes it significantly worse. So there's so many complications that come with drug creation and pharmaceuticals, which is why big pharma, while we always talk about how they make a bunch of money, a lot of the money actually tends to go back into drug trials. Of course, there are the big hedge funds at the top that make a lot, but a lot of the time for like the average researcher, right, a lot of the money just that is made from big pharma just goes back into clinical trials. So clinical trials are expensive. They're really expensive. So yeah, we talked about a lot of these drugs already. So Taxol, colchicine, colsamide or colsamide, vincristine, vinblastine, nicotazole. These are really, really important drugs because, if you see here, we have Taxol, which prevents microtubule depolymerization, so your microtubules can't, break down. And we have things like vincristine and nicotazole, which actually cause them to depolymerize. So there's a lot of things I hear that break division abilities, so... Already talked about these in one of my first couple lectures, so I'm not going to talk about them too much. What you should really know about them is Taxol, prevent microtubule. The rest of these, cause. Important to know. There are many viruses that are associated with cancer. Some viruses cause cancer, and so this is just something to be very aware of. So like hepatitis B, I think, is on here, right? Yeah, hepatitis B causes liver cancer. There's a bunch of these Epstein-Barr viruses, human herpesvirus. There's a bunch of these that cause cancer, so you guys got to be really careful with these. But yeah, this is actually the part of the lecture where I haven't learned this very super well, but I'm going to try my best to kind of go through it and explain it. But viruses, like I said, can cause cancer, but viruses are interesting because the way that they would cause cancer is through the proteins that they produce. So the proteins they produce must be cancer-causing. So we have a papillomavirus. I always mess this name up again. There's so many names that I keep messing up. Papillomavirus. I'm just going to call it the pop virus, I guess. I don't know. Yeah, so at least it's cervical cancer, but... so this virus is a double-stranded circular chromosome, so it's a plasmid, that has 8,000 nucleotides. So it's actually really small in comparison to a human genome, which is like 3 billion base pairs, right? So this is really small. So... chromosomes, right, are normally stably maintained in the basal cells of the epithelium as closed circular plasmids. So basically what they're saying is this plasmid is stable. So, yeah. What they're trying to get at here is that the replication process of this viral DNA is actually kept in very close proximity and timing with the host chromosome. And so what this means is that the virus itself only actually messes with the host cell cycle when the virus is programmed to produce certain infectious proteins. And so basically what this means, it's a dormant virus, right? So if it basically sits there and replicates over and over again until eventually it gets a signal to start producing the proteins that cause cancer. And here is a, interesting thing. Oh yeah, one thing to keep, this right here is generally harmless. It is supposed to be harmless. It does not do anything unless you integrate this plasmid DNA into your DNA. It usually is harmless, right? So it says relatively harmless, right? However, there are cases where the integration of a fragment of this viral DNA can disrupt normal control of the gene expression, right? So you can see here, rare accidents can cause the integration of a fragment into the chromosome of a host, which alters the environment of the viral genes in the basal cells of the epithelium. So these viral genes are now exposed to probably more transcription, more translation. And this disrupts the normal control of viral gene expression. So normally these viral genes are expressed at some level, whatever. But then when they're in the actual chromosome, they might be expressed a lot more than they normally are. And so the unregulated production of certain viral proteins messes with cell control and cell division, and therefore actually causes cancer because it leads to proliferation a lot of the time. This is heavily dependent on where the virus actually inserts itself to. But the main thing to keep in mind is here that when you insert yourself into the host genome, you have a lot more access to some of the host genome proteins than maybe a smaller plasmid would, because it's just probability, right? It's much easier to find a much larger floating 3 billion base pair molecule than an 8,000 base pair plasmid, so. HPV vaccine, yay, 2006. But yeah. Vaccines have been really helpful for bringing down rates of cervical cancers. That's always fun. There's also CAR T cells. Yeah, these are really interesting. Basically, what's happening here is that you're basically taking a T cell from a patient, and you are manually manufacturing T cells that have receptors for these cancer cells. So you take a T cell from that patient, you make these CAR T cells, so you insert genes for that code for proteins from the cancer that you're looking at, and then your T cell will make receptors for that, and so you grow millions of these, and then you infuse these into the patient's blood. And so these T cells that you just made with this DNA will start binding to these cancer cells and destroying them because because these receptors are able to pick up one of the antigens on the cancer cell surfaces. Pretty cool process, actually. Oh, and this is just a nice little diagram of what you should know for your exam. Yeah, I'm not going to harp on this too much because you guys have already seen me talk about a lot of these in my previous videos. I will scroll through them real quick and maybe talk a little bit about what you should focus on maybe, but not really too much from me. So, already talked about this. Just remember this table. This table is really useful. Just put this table on your note card. Really helpful, super easy to understand. Great. Remember, for actin, grows and shrinks. When ATP is bound, it's really happy to bind to other actin monomers. When it's not bound, it doesn't bind very much. yeah, so ATP high affinity, ADP low affinity. So it grows from the plus end, generally speaking, shrinks from the minus end, right? For this, we have... Oh, yeah, remember these terms: catastrophe, rescue, GDP cap, and that. How actin has its role in muscles. Remember that actin is important in cross-bridging, so this is the whole cycle here. Something that you can maybe put on your note card if you want. Super helpful, I think. Kinesin and dynein, remember, how do they walk? Remember the mechanism of hydrolyzation, and then movement, and then attach another one here, and then keep going, right? Just a good process to remember. Regulation of CDK. Remember that the tyrosine one is... the tyrosine one is is inhibitory, and the threonine one is activating, right? So this is something to remember. Or sorry, no, I think I mixed that around. Sorry, the tyrosine one is oh no, sorry. The tyrosine one is inhibitory. I always mix these up. I don't know why I mix these up. I believe they're inhibitory, at least. I feel like I'm messing this up. But yeah, so the tyrosine one here, I mean, just remember these two phosphate groups. Don't quote me on that, because I think I messed that up. But just remember that it's like a double fold mechanism, right? Where the threonine here stops it from entering the cell cycle, basically. And so the addition of this tyrosine actually allows it to... it basically acts as another safety mechanism, if you will. So just remember that. This pathway, honestly, probably worth putting in your note card. I genuinely cannot just say that this is easy to memorize. If you feel like you can memorize it, great. But no shame putting on your note card. Use your note card as much as you can. Super helpful tool. And here is the APC, the anaphase promoting complex that I was talking about earlier. So cyclin targeted for ubiquitination, degradation by anaphase promoting complex. So remember that cyclin is degraded by this. So cyclin or APC is actually a tumor suppressor because it prevents proliferation. Talked about this, the cell cycling path. So cell cycle, remember RB and the E2F pathway. Remember that RB is a repressor for E2F. And when phosphorylated, RB leaves and E2F promotes cell division. RAS is from the external growth factor pathway, right? It's the G protein, acts as a JEF, and then, hydrolyze the GTP to activate the MAP kinase pathway. And then P53 is the guardian of the cell, right? Goes through that process with MDM2. So this is just something really important. And remember, P53 is a transcription factor. So, yeah. Talked about hedgehog. Remember that patched is going to be a tumor suppressor, and smoothened is going to be a proto-oncogene, right? So smoothened promotes activation of this, and then patched stops it from doing that. And here is a image of Wnt signaling as well. Wnt, remember, is a proto-oncogene as well, and things like APC would be the tumor suppressors, right? And here's this nice little diagram again, but other than that, that's actually going to be the end of this video. I hope it was helpful. Again, please feel free to leave comments if you have questions or if I didn't explain something super well. I'm like getting through the senioritis at this point, so I feel like my videos aren't maybe as good as they should be, but we're going to try our best to get through this, and maybe at least if they're a little bit helpful for you guys and maybe I explain something a little bit easier for you to understand, that's a win in my book. So I hope it was helpful. If it was, great. If it wasn't, oh boy. But yeah, have a good one, and happy studying.